Pag-fix ko na to. Balik ang index card.
Ito yan, guys. Ito yung aking SBS.
For one and two, that's only a maximum of two sentences, minimum of two sentences. It could be three words. It could be three words. Eyes on your paper. Eyes on your paper.
Chapter 7. Photography. Thanks. Wala sa slides.
So how do you differentiate reductionism and holism? Sabi ko pwede 3 words lang. You can just say part versus whole.
Yay.
Okay. Next, how about mechanism and vitalism? So that is you are dissecting components versus that vital force. Yep. Identify the 2 strains used by Frederick Griffith in his transformation principle experiments and indicate which is pathogenic and which is not. So you have the smooth and the rough type. The smooth type is the virulent, the pathogenic. The rough type is the virulent or non-pathogenic. Now we have number
Oh. We have number 4. So the isotope used by Hershey and Chase for labeling T2 phage DNA. So this one is that is P32, and for protein that is S35. Now for number 6, name the important discovery or experiment associated first with Hershey and Chase. What is this? It's what kind of experiment? That's the blender experiment. And for Frederick Vischer, that is nuclein. And Erwin Chargaff, huwag yung ilagay na Chargaff's rule. Okay? Anong Chargaff's rule yun? So that's the ratio of purine and pyrimidine, which is 1:1. 1:1 ratio, purine and pyrimidine. Yeah. Then number 7 being the crystallographer and her famous image of DNA that allowed Crick and Watson to elucidate the double helix structure of DNA. So that is, who is this? Rosalind Franklin, and the famous photo is photo 51. And number 8, what's this? So it is the study of genes and its products and how these products function and interact in the organization and perpetuation of living things. So if your answer somehow plays there, okay na. Pero pag completely tama, well, you get a full points. Okay, that's like 2 points. If not, mga 1.95. Sige. Okay, so that's all for quiz. I will return your paper next meeting. Ako na yung mag-check nun. Okay? So, let's start our lesson for today. So, nandito na tayo sa lecture 2, nucleic acids. So, para lang siyang review nung previous nyo na pinag-aralan from Bio 30 and even siguro sa Chem, you already tackled this. Sa Biochem, meron ba nito? Meron? Yun. So, very easy na lang to sa inyo. Parang, review lang. So, for the learning outcomes for this lecture, so at the end of this lecture, you should be able to first describe the components and structural features of nucleotides and explain how nucleotides are linked together to form polynucleotides and discuss how DNA is packaged in the nucleus of the cell. So first, let us have a review of what is a nucleic acid. Okay, so nucleic acids are known to be the chemical repositories of genetic information. So this is where genetic information is stored. Now, it is located inside the nucleus for eukaryotes or in the nucleoid region for prokaryotes. And also known to be biopolymers of nucleotides are linked together by phosphodiester bonds. So later on, we'll see that in the structure of your nucleic acids. So it has 2 forms. You have your DNA which is your 2' deoxyribonucleic acid, so that's the full government name of your DNA, and RNA, which is ribonucleic acid. Alright? Now so this is how it looks like. So your eukaryotic cell, so DNA is primarily housed inside the membrane-bound nucleus. Okay? So whereas in prokaryotic cell, meron siyang main circular DNA. So that is inside or found in the nucleoid region. And it also has this smaller plasmids, which is an extra chromosomal circular DNA that can carry non-essential genes for advantages like antibiotic resistance of that certain organism like bacteria. Now let's go to the main topic, which is the nucleotide and nucleosides. So ano yung pinagkaiba ng nucleotide sa nucleosides? Tides and sides. What's the difference? What's the difference? Your nucleotide is composed of, just a review, composed of how many components? 3. That's right, Archie. You have first the phosphate group, the sugar, the pentose sugar to be specific, that's the ribofuranol sugar, and then the other one is the nitrogenous base or the nucleobase. What about your nucleosides? Anong meron sa nucleosides na wala yung nucleotide? It has no, wala siyang phosphate group. So yung component niya is just the sugar and the nitrogenous base. Now let's take a look at the structure of your nucleotide. Can you give me an example of a very important, or one example of nucleotide na very important, especially in energy metabolism? Yes, Jewel. Luis. Yes, ATP, adenosine triphosphate. So it's known to be the universal energy currency, but we also have this another form of energy currency, which is very specific when we go through the process of translation. Ano kaya yun? Hindi ATP yung ginagamit sa translation eh. Ano siya? It is? Ano ginagamit na energy sa translation? That is also a nucleotide. GTP, that's right, it's GTP. And we also have other structural moieties of coenzymes that function din dyan ng nucleotide. Are you familiar with NAD plus FAD? Ayan. Nucleotide din yan. So let's take a look at the importance of this. Kasi ito ay also your precursor of your building blocks of nucleic acids. Now, let's go to the first part or the first part ng ating nucleotide, which is your pentose sugar or pentoses in nucleotides. So this one is what you call a furan ring. F-U-R-A-N, furan ring. So it is known as furan ring because this is a 5-membered aromatic heterocyclic compound. Okay? And notice, in nucleotides, na pag-aaralan natin, so may 2 forms tayo, di ba? We have the DNA and the RNA. So one of this sugar here, or pentose sugar, is beta D2' deoxyribose, which is a sugar for DNA, and the other one is the beta D ribose for RNA. Now, why is it called a beta sugar? Why beta? Bakit siya beta? Again, ang sabi ko kanina, this is aromatic. It's a ring. And you notice, nasan yung carbon 1 niya? This is the carbon 1. So this is also known as the anomeric carbon. This is an anomeric carbon in carbon 1, yung kanyang structural moiety is pointing upwards. This one is hydroxyl group. So that is beta form. Pag nasa baba siya, that is alpha form. Next, bakit siya tinawag na D? Now, it is D because the OH or the hydroxyl group is to the right. This is right and this is, sorry. No. Yeah. This is to the right of your in Fisher projection. So your penultimate carbon, the second to the last carbon, yung hydroxyl group niya, nasa right, kaya siya nasa D4. Pag nasa left, yan ay nasa L4. So since nasa right siya, it is a D, in D form. Kaya siya beta D. Okay. Next naman, bakit 2' yung DNA? It's because in the 2' carbon, wala siyang oxygen. So, kaya sa deoxyribose. 2' deoxyribose. And for an RNA, that is a ribofuranose, so that is a ribose. So, completely okay po siya. Ayan. Okay na? Okay. So we know why is it named beta D2 prime deoxyribose and beta D ribose. Now let's go to nitrogenous base. So that's the next component of your nucleotide. So ito yung overview ng nitrogenous bases natin that is incorporated in both DNA and RNA mostly kasi may other nitrogenous bases pa rin, not only this. When we encounter the tRNA, meron tayong methylated mga bases. So, hindi muna yung cover. Ito lang muna yung cover natin, which is the two classes of nitrogenous bases. You have your purine and pyrimidine. So, what are these nitrogenous bases? So, notice muna, when you go, take a look at here, your purine has two big rings. So it's bigger compared to your pyrimidine, which has only one ring. So notice na meron ka conjugated bonds, and it is made up of heteroatoms. When we are talking about heteroatoms, it is not only composed of carbon and a ring, but it's alternating carbon-nitrogen atoms. Kaya siya tinawag na heterocyclic aromatic compound. Next. Also, you can see here, carbon 9. See the carbon 9 here? The carbon 9 is where your base is attached to the carbon 1 in your pentose sugar. So ito yung naka-attach sa pentose sugar, that nitrogen 9. So, this one for adenine. Remember that? For your purine, ang naka-attach naman for your sugar is your nitrogen 1. Very important to kasi dito natin ma-identify yung actual linkage niya later. Okay? Remember, for purine, this attaches to carbon 1 of your sugar in the nitrogen 9. For your pyrimidine, that's nitrogen 1. Okay? Now, let us review what are nitrogenous bases. First, again, derivative siya ng purine and pyrimidine. When we're talking about derivatives, it comes from purine or pyrimidine because you guys are derivatives of your parents, and likewise, this one, these nitrogenous bases are derivatives of either purine or pyrimidine. Again, I've mentioned that it is a heterocyclic aromatic compounds because it contains heteroatoms. Not just purely carbon, but it's alternating carbon-nitrogen atoms. Also, it is basic. What's basic? Hindi yung basic na easy lang siya, ha? It's basic because it's the presence of nitrogen with lone pair. So no that, remember you have your Bronsted-Lowry bases, okay? So this is considered because they are known to be proton acceptors, okay? Next, it has conjugated double bonds. What is the importance of these conjugated double bonds? What's the importance? Resonance, yes. Another, in quantification of your nucleic acids. Okay? Because your conjugate double bonds absorb UV radiation at 260 nanometers. Okay? So this is also a way for you to quantify your nucleic acids when you work on nucleic acids, of course. And then it has multiple sites for hydrogen bonding. Very important yan kasi it forms hydrogen bond. Yan yung nagko-connect sa dalawang polynucleotide ng DNA double helix. Okay, very important na meron siyang hydrogen bond kasi that connects your nitrogenous bases. Pairing. Okay, that's the pairing of your adenine, thymine, cytosine, and guanine. Okay? Ito pa pala. We will name this, what do you call this? This nitrogenous base, but sa kabila. Okay. Now, the mother ring, ni adenine, siya ay purely purine, tama? So, notice na yung kanyang 6 carbon ni adenine, nakita mo yung 6 carbon niya? Ano yung naka-attach? This is what group? Amino group. So, adenine is also called 6-amino purine. Next. How about guanine? What's the root word here is purine yung last niya. So, sometimes try to name guanine. What? So, carbon, anong carbon to? Carbon 6, meron siyang oxo group. Sa carbon 2, meron siyang amino group. So, anong tawag sa kanya? 2-amino 6-oxo. Ang nakalagay dyan ay oxy, pero oxo. 2-amino 6-oxo purine. Okay, who can try for cytosine? This is, kanina, purine. Yung sa baba ay pyrimidine. So, notice sa cytosine, yung carbon 2 niya has oxo group. The carbon 4 has amino group. Ano tawag sa kanya? What's the government name of your cytosine? Okay. Nice. Close. Pag nag-name tayo, unahin natin by alphabet tayo. So, uunahin natin si 4-amino 2-oxo-pyrimidine. Next, thymine. So, meron kang oxo group sa carbon 2 and carbon 5 meron siyang methyl group. Okay? So, ano tawag sa kanya? 5-methyl 2,4-dioxo-pyrimidine. Dioxo, kasi dalawa yung kanyang oxo group sa carbon 2 and carbon 4. How about uracil? Uracil is? Yes, that's 2,4-dioxo-pyrimidine. Ano yung pinagkaiba? Diba si uracil is only present, naturally present sa RNA. Your thymine is naturally present sa DNA. What's the difference of thymine and uracil? Yeah, your thymine has methyl group sa carbon 5. Kaya yung other name ni thymine is 5-methyl uracil. It's also known as 5-methyl uracil. Is it the same yung ginagamit niyo sa chem?
Wala akong
Hindi. Hindi niya name. Okay, sorry. Hindi pala siya pinapangalanan doon. Alright. Okay. Dioxo. Hindi dioxy. So, yan niya na. Pakicorrect na lang po. And then, let's go to different tautomeric shift in bases. So if we have what we call tautomerization, so this is the rapid interconversion of your isomeric forms. So if you have a pair of tautomers, yun yung dalawang isometric compounds or structural isomers of each other. It means that they have the same molecular formula, it's just that meron silang different atom-to-atom attachment. Okay, we have first here your amino-imino forms of your cytosine and your ketol-enol forms of guanine. So bakit importante na malaman natin yung anong pinaka-stable dito? The amino or the amino form? Amino or imino form? The stable form? It's the amino form. How about the ketol and enol? Which one is stable? More stable? It's the ketones. But then it's still the rapid interconversion of your isometric or structural isomers of each other. Now, why is this important? Because when we have tautomeric shift in your bases, these tautomeric forms can result to what we call anomalous base pairing. Kasi mag-iiba yung atom-to-atom attachment, di ba? So, mag-iiba din yung available na lone pair, yung proton acceptor or yung proton donor. So, mag-iiba yung pairing nila sa what do you call this? Or formation ng hydrogen bonding. So, for example here, you have your thymine, that is the enol form, and it pairs with guanine in its keto form. So, nagkakaroon ng anomalous base pairing. So, thymine normally pairs with what? Adenine. But in this case, it pairs with guanine in its enol form. For cytosine and adenine, they pair together because your cytosine is in imino form. Okay? So if it is in its amino form, your cytosine will form bonds with guanine. However, in this case, there is a different, there are changes in the atom attachment, hence it forms bond with adenine. Okay? So this is why it's very important kasi this tautomeric shift, this anomalous base pairing can lead to what? Yes, it can lead to mutations. Okay? Next, let's take a look at how nucleotides are formed. Masakit ba sa ano to? Malakas. Okay. So, formation of nucleotides. So, in order for these nucleotides to form, it has to liberate a molecule or two molecules of water. So, take a look at this sa taas. If this will be liberated, that will form water, and this also will form water. Two waters will be liberated to form that nucleotide. Okay? Now let us review the three main linkages of your nucleotides. The first one is what we call the phosphoester bond. So that is the bond that connects your sugar, your pentyl sugar, and the phosphate group. And the other one is your beta-N glycosidic bond. Now, in your Bio-30, you just call it glycosidic bond. But here, it has to be specific to be the beta-N glycosidic bond. But for bonds of purine and pyrimidine, may specific pa rin doon. So, specify pa rin natin. Okay? We'll go through that later. Now, tingnan muna natin ulit si nucleosides. We mentioned this earlier na yung nucleosides does not contain phosphate group. It only contains your sugar and your nitrogenous base. Now, it's sugar and base only, and this one carries the genetic information because you have different sugars for DNA and RNA, and you also have different bases. Alright? Now, for base na adenine, if the sugar is ribose, that's for RNA, it's called adenosine. So may tatlo kang phosphate na dinagdag dyan, magiging adenosine triphosphate siya. Now, for if it is has a beta 2' deoxyribose, that is called 2' deoxyadenosine. For cytosine, that's cytidine for RNA, 2' deoxycytidine for DNA. Guanine, guanosine, and 2' deoxyguanosine for DNA. And thymine, that's 2' deoxythymidine. Wala namang thymine sa RNA, so that's none. Uracil is just uridine, and there's no uracil in DNA, normally in DNA. Walang uracil. Now, let's go to naming nucleotides. So how do we name it? So you have your nucleoside, yung kanina, sabi ko, have here adenosine for your RNA. And if you add 3-phosphate group, it will become adenosine triphosphate. Okay? So, say for example here, in this illustration, so you have your sugar, 2' deoxyribose. Yung base niya is adenine. So, if meron kang isang phosphate group, ang tawag sa kanya ay 2' deoxyadenosine 5' monophosphate. Kasi siya ay isang phosphate lang, monophosphate. Pag dalawang phosphate, diphosphate, tatlong phosphate, triphosphate. And this one is what you call a 2' deoxyribonucleotide. Okay, so this is one form of your nucleotide, 2' deoxyribonucleotide. The other one, if you have a sugar that is a ribose and meron kang base na cytosine, this is what you call cytidine 5' diphosphate. Because you have how many phosphate groups? You have 2 here attached. So that is a CDP, and this one is not a deoxyribonucleotide but a ribonucleotide, the other form of your nucleotide. So you have your two forms introduced to you. You have your 2' deoxyribonucleotide and the ribonucleotide. Next, let's go to the phosphate group, which is very important. So this one forms the backbone of your polynucleotide together with the pentose sugar. So, yung nasa labas ng your DNA backbone is your sugar phosphate backbone. Inside it are the nitrogenous bases, right? So, your phosphate group is very important because this one imparts the overall charge of your DNA, which is negative. And that negative charge is very important, especially in DNA packaging and how your 6' DNA can fit inside that very tiny nucleus of yours. Okay? So this is very important that you remember the phosphate group is the one that imparts the overall negative charge of your DNA. And again, this enables your DNA, this negatively charged phosphate, associate with your positively charged histones. So later, we'll see how your DNA is packaged inside your nucleus by associating it with different proteins, especially histones. Okay, now let's take a look at specifically to deoxyribonucleic acid. So dito muna tayo. Now, the primary structure of your DNA, so this one is a linear sequence of your deoxyribonucleotides, and it has two complementary strands held together by hydrogen bonds. So remember that your DNA is double helix, and each one of that side is a polynucleotide, and they are attached together by what you call hydrogen bonds. Alright? So the backbone is alternating sugar phosphate groups linked together by what we call phosphodiester bond. This phosphodiester bond is not a is a covalent bond. Okay, this one is a covalent bond. Okay, so this is the primary structure of your DNA. So you have here your DNA polymerase. So you have your incoming DNTP or deoxynucleotide triphosphate that is attacked by the alpha phosphate by the 3' hydroxyl of your growing DNA chain. So we'll further discuss this when we go to DNA replication. Now, so this is the primary structure of your DNA. So parang nagre-review lang tayo ng Bio 30 dito. So remember na yung naming pala ng carbon for your sugar, meron siyang priming. So 1', 2', 3', 4', 5', ganon. This is done especially to differentiate it from the carbon found in your nitrogenous bases. And very important din na naka-prime siya kasi this is the convention of the direction also of your DNA strand from 5' to 3'. Because in the 5' end, that is where, this is where your, naka-attach sa 5' end? Phosphate group. So in the 5' end of the 5 carbon of your pentose sugar is where your phosphate group is attached. At yung nasa dulo is your 3' end. The 3' end, this is your 3' hydroxyl end. So this one exhibits the nucleophilic attack to the incoming DNTPs or deoxynucleotide triphosphate in the growing DNA strand. Okay, so this one is the one that does the nucleophilic attack. So what bond is this? That's the phosphoester bond. How about this one? The beta-N glycosidic bond. Now, if this is a purine, you call that beta-N9 glycosidic bond. Because where does it attach? In the nitrogen 9. Kaya siya tinatawag na beta-N9 glycosidic bond for purine. Now, what if pyrimidine yung naka-attach? Ano tawag sa kanya? Hindi na siya beta-N glycosidic bond, but it is beta-N1 glycosidic bond. Because it is a pyrimidine that is attached to the sugar. Follow? Now, nasaan dito ang diester bond? Diester bond links the two adjacent nucleotides. It links the two adjacent nucleotides. So, this one, hindi siya equivalent. It's non-equivalent phosphodiester bond because one is longer and the other one is shorter. Kaya meron kang proximal at distal bond. This is a distal bond, mas mahaba siya. This is a proximal bond, mas malapit siya, mas maikli. So your phosphodiester bond is two non-equivalent phosphoester bond. Alright? So the convention or the direction of your DNA is always, the growing strand is always 5' to 3'. Okay? Alright. So let's take a look at the example of the bases of our DNA sequence when you do your analysis if you're working with DNA. So by convention, the base sequence is read from 5' to 3' direction, and the base sequence along a chain carries the genetic information. So example for this is your amylase gene for Homo sapiens. Take a look at that. It's just letters. So what is the significance of these letters? These are nitrogenous bases. So you imagine this. This is 5 prime. It starts with guanine as a base. So since this is DNA, you assume that this is a alternating yung kanyang sugar phosphate backbone, the sugar here is 2 prime deoxyribose. So this is your last base here, which is adenine. So you just imagine it's a stretch of our long DNA, tapos ito yung sequences niya. Okay? Nagkakaiba-iba lang sila sa nitrogenous bases. They have similar phosphate sugar backbone, kaya it doesn't make sense kung ilalagay mo pa siya. Okay? Just the nitrogenous bases. Next, let's take a look at the secondary structure of your DNA. Now, normally, your DNA is double-stranded. Normally. May single-stranded ba ng DNA? Does it exist? Yes, it does. It happens. So your DNA molecule has a form of a double helix, which is a 2 intertwined antiparallel strands of nucleotides. Bakit antiparallel? The one runs from 5' to 3', and yung complementary niya is 3' to 5'. But the direction, of course, is still 5' to 3' in the growing chain. Okay? Now, your DNA has major and minor grooves. So why is it important? So your major groove, yung distance ng phosphate backbone is malayo sa isa't isa. But for the minor groove, it's closer. That's how you differentiate the minor and the major groove. So your major groove is very important, especially for binding with important transcription factors, also for gene regulation. We'll further discuss that when we go to transcription and DNA replication. It's also very important kasi yung major grooves can be easily accessed by your polymerases, and your sequences are naturally, kumbaga, exposed siya. Most of the sequences are exposed. So can be easily accessed by transcription factors or polymerases. Now, this major and minor grooves is only true to bDNA. You know that, di ba alam niyo naman na meron tayong different conformations ng DNA. Hindi lang bDNA, meron tayong A, meron tayong Z, C, E. Ganon. So, this minor and major groove is only true for bDNA. Only true for bDNA. Okay? Aside from that, most of the DNA helix natin, meron para siya tinatawag na orientation. It could be either right-handed or left-handed double helix. So most DNA in nature, especially bDNA, is right-handed. They are right-handed helices because they rotate clockwise. And you have a few that rotates otherwise, counterclockwise. They are called left-handed helices. And the known left-handed helices are what you call the zDNA. The zDNA is known to be a left-handed DNA. Now, aside from that, for the secondary structure of DNA, we have important na features niya is the base pair stacking. So this one maximizes the hydrophobic interaction for greater stability. So that is around 3.4 nanometers. So the distance, not 3.4 nanometers, but 3.4 angstroms. So that's the distance of the two base pairs, or 0.34 nanometers. So the distance between this and this adenine and cytosine here is 3.4 angstroms. Okay? And the constant diameter of your DNA helix is around 20 angstroms or 2 nanometers. And this one is maintained by the right pairing of your bases or the specific base pairing. Okay? So this one, just a review lang naman. So adenine pairs with thymine that forms two hydrogen bonds. Guanine and cytosine forms three hydrogen bonds. Now let's take a look at the different DNA conformations. So meron tayong DNA A, B, C, D, E, and Z. But the ones that we'll discuss today are the three common ones in nature, which is DNA A, DNA B, and DNA Z. So this one, sabi ko kanina, yung major and minor growth is normally or only true for your B DNA. Because notice, na yung minor and major growth is very prominent. Pakikita mo talaga na meron siyang minor and major growth. So your A form naman, still right-handed, pero notice that it is more bulkier and wider compared to your B form. Your Z form, on the other hand, is narrower compared to your B form, and this is left-handed. Okay? So this is the other illustration of your different DNA conformations. Most likely, ganito yung structure ng ating zDNA because of its alternating GC component. Marami siyang GC component, and this actually exists in high salt conditions. Okay? Alright. So let us differentiate the different DNA conformations. So for the helix type A, the overall proportion is short and broad. For B, that's the normal one that commonly occurring, it's long and thin. C is also long and thin. However, for Z, it's left-handed. It's elongated, narrower, and slim. A to C, yung rotation ng helix are all right-handed. However, for Z, it's left-handed. For the base pairs per turn, A is bulkier, so it's wider. It's around 11 bases per turn. For B, DNA B, that's around 10 bases per turn. For C, that's 9.33 bases. And for Z, since it's mas mahaba siya, it's around 12 bases per turn. So for the conditions of occurrence, DNA A is 75% in human, and then these are the possible ions present around that DNA. Now, yung DNA A typically exists when we have what we call DNA-RNA hybrid. Usually, nagkakaroon tayo ng DNA A form with DNA-RNA hybrid, and aside from that, your RNA, when it forms, your RNA can form a helix. So your RNA can form a helix, and yung form niya is DNA A na form. So it's bulkier, and it's wider than the DNA B form. Okay? Hindi siya, DNA, it's RNA. So your RNA niya is in A form. It's not DNA A. The RNA, if it forms a helix, it is in A form. Sorry, medyo nalito kayo. Kung yung RNA, nagpo-form siya ng helix, nasa A form siya. Yun, tama na. For B, it's 92%, usually for low salt condition. DNA C is 66% in human. For Z, it occurs in high salt condition. Di lang nalagay. It occurs in high salt condition, and marami siyang GC content. It has a lot of GC content. In humans. It present sa human. If you isolate or study in humans, 66% you can find 66% of the time you can find is DNA C. conformation of the DNA. Now, the helix diameter, so mas malaki or mas wider yung A form, so that's 23. B form is the normal one, we have the 20. C form is also 20. And Z form is narrower, so that's 18 angstroms. So, for the major group, it's extremely narrow and very deep. For B, that's wide and intermediate. For Z, it's flattened out on the helix surface. So yun lang lahat yon. When we are talking about A form, ito yung sinasabi ko kanina, it exists most likely in RNAs. Nagkakaroon tayo ng A form sa RNAs. So it's found on your double-stranded RNA. Your double-stranded RNA can form a helix. It is in A form. Also, RNA-DNA duplexes or DNA-RNA hybrid. Meron kayong A form dyan. Your RNA, tRNA, double-stranded RNA, viral RNA, and polypurine strand can form an A-form helix. So your B-form helix naman is biologically dominant, and it's polypyrimidine or polypyrimidine strand. For C-form, in vitro, that is present usually in dehydrated conditions. And for DNA-Z, ito, meron siyang alternating cytosine guanine sequence, and it is present in high salt condition, and although in vitro pa lang siya although in vitro pa lang siya napag-aaralan pero yung function niya in vivo is not yet fully described, yung Z form. Alright, next. We have your DNA supercoiling. So, ito yung last slide ko for today's lecture. So, your supercoiling actually, this one, refers to the extra coiling of your double-stranded helix on the top of its normal helical structure. So, meron tayong tinatawag na positive or negative supercoiling. So, if your DNA is overwhelmed, so may tension, imagine a rope, tapos in-overwhelm mo siya, or binaliktad mo siya, it's still what we call, it's still supercoils in other way around. Pag if you move it, say for example, clockwise direction, pag clockwise direction yung DNA, if you move it on the clockwise direction na extra movement, that will coil it positively. Pag you coil it alternate, example, your DNA helix moves clockwise and you twist it counterclockwise, that will make it negatively supercoiled. Okay? So, if your helices moves clockwise direction and you twist it extra more, another clockwise direction, and hindi supercoiled siya, it means positively supercoiled. But if you move it opposite, it will be still supercoiled in a different way, but it is what we call negative supercoiling. And all organisms store their DNA as negatively supercoiled. Bakit? This is actually for DNA gene expression, sa gene regulation, ganon. However, the problem when it is negatively supercoiled is that it decreases storage space. It occupies a lot of space kasi kalat siya. And then, even with that, it's very conducive for what we call replication and transcription factors. Yung positively supercoiled kasi, let's say for example here, when you are increasing supercoiling, nagiging positively supercoiled siya, mas lumiliit yung kanyang space. I mean, it occupies less space in the cytoplasm. Are you familiar with, di ba nag-gel electrophoresis kayo sa chem? Ano ba? Hindi. ML lang pala ako. Okay. Kasi may sasabihin sana ako na ano siya. Let's say for example kasi, if your DNA is normal lang pala, hindi pa siya supercoiled, the normal this one. If you run that in gel,
malaki kasi yun siya. It takes space. So, nasa taas lang siya. Pag siya ay super coiled, mas mabilis siyang bumaba. Okay. So, because it takes up less space lang din. Ito. Ayun. Now, for your assignment, 1, 2, 3, 4, 5, 6, 7, 8. Ang assignment nyo, you will have a short reporting. Short lang ha, huwag yung occupy yung buong lecture class. You can find this journal online. Lahat ng sagot sa i-report nyo nasa journal lang na to. Ang journal, title niya is A Bouquet of Dairy Structures, Emerging Diversity. Yung nakalagay yung title ng journal. Hanapin nyo to, basahin nyo, and then you will present this one. Now, mag-group ako, so inisip ko kung paano ko kayo i-group. Dali lang. It's a journal article. Now, ganito na lang. I will group you by row. Sorry. This is group 1. Something na kayo nakapag Group
Pinagtayo tayo.
Group 3, 4, 5, 6, 7, 8, 9. Now, kayong tatlo. Wait lang ha, kayong tatlo. Ikaw, dito ka sa kanya. Dito kang group. Ikaw, sino yung tatlo yung group? Yung tatlo. This one. Mag-group ka dito. Oh, group 2 ka. And you, the last one, group in here. 1, 2, 3, 4, 5. 5. So, you are group 9, group 2, group 5. Okay? So, next meeting, meron kayong tinatawag na 3-minute presentation. 3-minute ha. Mabilis lang to. You can make a infographic na nandun na lahat, tapos sabihin nyo na lang siya. Hindi yung, you do not take so much time ha. So, when are these formed? Paano tayo nagkakaroon ng cruciform DNA? Bakit nagkakaroon tayo ng changes na ganito? And bakit importante na meron kayo ganito? What's the importance of this? Different structure of DNA. So, surprisingly, your DNA is not just a double helix. It has a lot of non-canonical structures. So, let's see later, next meeting, kung ano yung